Auzubillahi Bismillahirrahmanirrahim. Today, my topic of this seminar would be on Pestis Despetis Ruminants and determination of risk factor associated. My name is Dr. Moazam Jalees and I am Assistant Professor in the Department of Microbiology. So, this PPR or Pestis Despetis Ruminants is a significant and important disease of small ruminants, sheep and goat. Mostly, they are very deadly virus and it is distributed worldwide. And most importantly, they are transboundary in nature. So that means all the neighboring countries, all the neighboring areas which are having this disease can easily be transferred across the boundaries of the nations. So this PPR, it is a commonly known as kata pseudo rinderpest pneumoenteritis complex stomatitis pneumoenteritis syndrome. So it's a highly acute and contagious and economically very important disease because it's a poor man's disease basically. And all the countries which are affected, the small holdings and the farmers, those who have 4 5 animals, small animals, and their livelihood is dependent on it, is mostly affected with this particular disease, and it is causing the huge economic losses as per these small ruminants are concerned. So the distribution is worldwide, as I've told you, and it is listed as list A disease by this OIE. Now we called it as World Organization for Animal Health. Previously, OIE was the previous name. So the listed A disease, it is ranked high among the list of diseases of the animals. And the distribution, regarding the distribution of the disease, it is worldwide. There are four lineages, reported lineages of this virus, and we are living here in this lineage 4 area, this Pakistan, where lineage 4 are most prevalent in this particular region of the Southeast Asia. And the distribution is, you can see here, the red dots. It is indicating this lineage 4 and lineage 2 with this 3 with this green, and lineage 1 and 2 accordingly. You can see here the change of color is represented. Now, uh, coming to the prevalence of PPR in different areas of the world, it was first time reported from the Ivory Coast in 1942, of the West African coast, and later is spread to the east side also, and later it was spread to this Asian region. And now you can see here from this 1942 picture to 2009, it is distributed all across the globe. All the countries are affected by it. So you can see here the prevalence rate is rapidly increasing since its origin back in 1942. Regarding the prevalence of PPR, it is mostly found in the areas of this Punjab, this Balochistan, and also Sindh, and obviously you can see here the areas of north sides are also affected by it. So no region is secure from this as per Pakistan areas are concerned. Regarding the reporting of this PPR, it was has been recognized with this typical clinical signs in which the diarrhea is there, in which the rapid increase of temperature and septicemia is the final outcome. So these symptoms, particular symptoms, are correlated with the rinderpest as seen in the large animals. But since its reporting in 1991, that was confirmed almost 2 years after that by Pervez, who conducted PCR at that time. and later by Athar also in 1995. So that disease initially was considered prevalent in the irrigated and non-irrigated parts of Sindh, northern parts of Punjab, few districts of Khyber Pakhtunkhwa, and southern areas of Azad Kashmir. But currently, the recent studies have demonstrated its prevalence in all five provinces. The endemic city of this disease, it's a major threat to about 114 million small ruminants of the country, causing huge economic losses with the current livestock census. So it is not stopping. These two countries in front of you, Greece and Romania, was declared this PPR free. But recently, the you can see the date at the bottom left. It is 14 August 2024. That means this year's few months back. So this disease, there are cases reported in these two regions, which was declared rinderpest free. So that means the countries, those who have declared themselves by passing on various criterias of declaring this PPR free, or again this disease of PPR is coming again to their lands also. Regarding the missing linkages of our study, we are focusing on the epidemiology because that's a continuously changing domain when you are studying a certain disease. So epidemiological picture might vary with the passage of time. You can't say until unless you fill out the criteria of this World Organization for Animal Health as declared as particular disease free, as in the case of this PPR. So previous reports are with different scenarios in Pakistan involving sheep, involving goat, although it is known as a goat plague. Sheep are affected, but not to that extent. But worldwide, sheep and goat are rated in the same uh, affected species of this particular PPR menace. So there is continuously outbreaks. There are continuously outbreaks. That's why I said that epidemiology is the continuously changing pictures or domain, and that continuously changing picture forced us to monitor this continuous repeated outbreak and check the viability of the vaccines if they are working or not. Regarding the virus structure, this virus is paramyxo virus, belong to this genus Morbillivirus, and it's an RNA having four lineages, and this genome is divided into six transcription units, and these six units are NPM, F, H, and L. This is hemagglutinin fusion matrix, phosphoprotein, large, and nucleocapsid and viral RNA is there. So that's a single stranded RNA having C and V non-structural protein with them. This is the virus replication mode. After the attachment with its spikes, it gains entry into the cell, and onwards it take the control of the nucleocapsids of the host cell, and finally it starts growing and replicating with the help of this mechanism, and finally transcription and translation of the this nuclear material goes on, and finally it got it released after and finally packed in the Golgi complex, same as the way the rest of the proteins got packed, and finally the assembly of the virus is there, and finally the progeny virus is released after bursting the cells out in the atmosphere to infect the other viruses. Regarding the approach to control this PPR, this World Organization for Animal Health approach is a stepwise approach. Basically, there are four stages or four steps which are involved in controlling this disease. And final, the fourth stage is declared, or it is declared PPR free when there is no virus circulation. So in our country by 2030, we have the plan to declare our country as PPR free, and there are multiple uh, steps which are going on with the government organizations and the government as well. So we are approximately at stage 2 or 3 in between. And these stepwise, if we see that OIE said that vaccine is the main tool by which we gain control of this particular disease, and it's the same in line with the treatment methodologies of the disease. And that's the way they managed it, is to provide the uninterrupted cold chain vaccines, and realistic estimate of vaccine quality is there side by side. In the first step of this vaccination program, there is implementation of the policies by which we take the control, and onwards, when the vaccine is there in the form of ring vaccine, in the present scenario, when the outbreak is there in Pakistan, we opted for the ring vaccine. But finally, the final steps, if before the declaration of the renders in this PPR free domain, the 100% of the small ruminants above 3 months of the age should be vaccinated, because the natural life is not more than that. The males got slaughtered well before that, just reaching the age of 1 year or before that. But females are retained for reproductive cycles for next couple of years, but not more than that. So as per the availability of life is there, after 2 years or after 2.5 years, there is no use of this vaccination. So Pakistan is moving and initiated its PPR control efforts in line with the global PPR eradication program that is launched by this FAO and WOA, this World Organization of Animal Health. Previously, we name it as OIE. So the lead agencies in Pakistan are National Veterinary Labs and Provincial Livestock Departments, and they are in 3 phases. They are operating. The first is risk-based control and vaccination. The phase 2 is surveillance and monitoring. And in the phase 3, they will go from an education and awareness campaign that goes side by side. And as I've told you, they declare it by 2030. But before that, in 2028, they are planning to get it attained that target. So this 2 year will be kept as monitoring and checking whether is there any outbreak or not. Regarding the risk factors of PPR in Pakistan, regarding the species susceptibility, goats are more affected than sheep. But if you compare here in this study by Muhibullah, he declared that sheep are got more prevalent, that disease is found more prevalent in that scenario. Higher prevalence, seroprevalence is more in 2 years, and obviously females are more affected as per serosurveillance because they are retained for reproduction. Peak outbreaks during the dry season, mostly in December, February, and April. Regarding the surveillance and monitoring, there are two methodologies, active and passive surveillance, in which we manage to get the data, and in our experiment, we adopted the same criterias. The first one is active approach, in which involves regular systemic collection of data through field visits and sample collection, and laboratory testing is simultaneously going on, and surveillance is there. The next is reactive approach. When farmers report that, or field veterinarians or field officers report cases, and we go on and we take the samples and different viscerals of sheep and goat. So we opted the same way. The objective of our study, we were thinking of conducting a sero-epidemiological study of PPR in Punjab province, and simultaneously we will go for this participatory epidemiological investigation. Regarding the molecular characterization of this PPR, we use F gene primers, And final this experimentation will be on the commercially available vaccines in Pakistan. Regarding the experimental design, we go for seroepidemiology with the help of C-ELISA kit and molecular characterization with the RT-PCR, and finally the vaccine evaluation. Zero prevalence for the estimation of sample size. This famous Michael Strasfield formula was applied, and expected prevalence in that we kept at 40%, and desired absolute precision is about 5%, and sample size was counted with that. So regarding the participatory epidemiology, we designed a performer, and on the basis of these performer, various interview were conducted, and finally the C ELISA competitive ELISA kits were imported from France. Regarding the molecular characterization, nasal swabs, blood, spleen, and lymph node were taken, and RNA extraction was done, and finally cDNA preparation. And this forward and reverse primers was taken with the 5 or 3 ends, and finally the RT-PCR with reference to the F gene with the specific primer were conducted. And finally, the phylogenetic analysis we performed. So, regarding the sample extraction method by triazole method was done, and cDNA was prepared, and finally the PCR F gene primer were taken. There's a master mix of this. You can see the cocktail here, and the thermal cycle. We opted for the isolation of this DNA, and finally the optimization of this PCR. Regarding the evaluation of vaccines, the first animal were taken as control, and there are 3 groups we made with 50 sheep and goat were taken in each group, and vaccines from VRA Lahore, Casvab, Quetta, and unvaccinated control, and the route of administration was intramuscular in all 2 groups, and unvaccinated control were taken as a control measure. And after that, we recorded the response after the immunization of these vaccine at 15, 30, and 45 and 60 days post vaccination. Results of the seroprevalence. It is more prevalent in sheep and goat, and you can see here it is more prevalent in goat rather than sheep in our study. District-wise seroprevalence, that was found more prevalent in Digikhan areas, and sex-based prevalence, it is more prevalent with the females in both in goat and sheep side. District-wise prevalence, that was more prevalent in the area of this Kasur, and age-related seroprevalence of PPR, it is more found seroprevalent in the sheep and goat aging more than 2 years of age. Seroprevalence basing on feeding pattern, they are more prevalent in open shed because they are more prone to the external environment in which mixing of the animals are there. So that could possibly be the reason. District-wise seroprevalence on the basis of feeding pattern, they are more prevalent in open land, but less in stall-fed animals. On the basis of pregnancy, this non-pregnant animals found to be more with this disease, and the antibodies are tighter are more found there. District wise, they are more prevalent in the case of this bhakkar and this pregnancy and non-pregnancy. On the basis of herd type, they are separate on mixed herd. So separate herds have in Digikhan got more prevalent, and mixed herd in other cases like Faisalabad got prevalence more in Faisalabad and Bhakkar region. Results of the molecular characterization regarding regarding using F gene primers, you can see here this 372 base pair, this ladder 100 base pair ladder was used, and 372 base pair of this viral DNA, this virus of PPR segments. You can see here the bands, clear bands are there with nominating 372 base pair signs. Regarding the district-wise prevalence of RT-PCR, we found more in the Bakka region than comparison to the other 5 districts we selected. And if we go for the different samples in sheep and goat, it is more found in the Faisalabad, in goat side, and also in Bakka region. And regarding the sheep, it is more found in the region of this Bhakkar also. This Kasur district is found to be more, almost 100% samples were found positive in that region. And same is the case with the lymph node. After this detection, we go for this analysis, sequence analysis. And for that very purpose, we use this phylogenetic analysis by neighbor joining method in the cluster W program. And we found this R region, and we registered that isolates also in the same gen bank sequence analysis. And we found that homology searching, it is more homologous with the region in the nearby region involving India, Bhutan, and Bangladesh. You can see here the arrow represents our reporting F gene, and it is more common with the neighboring countries and neighboring areas, and in the Pakistan region also. Regarding the results of the vaccine evaluation, these 2 commercial vaccines, you can see here the graph. It is found equally good, both of the vaccines, as the titer goes up and up to this 60-day post-vaccination. And group C is the control. The next 2 groups, it is almost the same. Regarding the conclusion of this seroprevalence, difference in prevalence between sheep and goat, non-significant was found. Young animals are more prone to infection. Female animals show significant higher prevalence. And there are open land grazing animals show significant higher prevalence also. Pregnant animals showed significantly higher prevalence, and estimated in current studies were higher than reported in the previous regional studies. Regarding the molecular characterization, predilection site was found high in the lymph node, as reported in the literature also, and the sequence study clustered with the sequence pattern from the Pakistan with Bhutan and Bangladesh by neighbor joining method. There is no significant difference between efficacy of the 2 vaccines as shown in the previous chart also. There are future concerns regarding this PPR disease that continuously require seromonitoring at the disease at country level, and characterization of the local isolates based on various epitopes, and vaccine development from the local and indigenous strain is the need of the hour. Thank you very much. That is all about my presentation.